class, today we're going to learn about the second line of defense, formed elements in blood. When microbes penetrate the first line of defense, they encounter a second line of defense that includes defensive cells, such as phagocytic cells, inflammation, fever, and antimicrobial substances. First, understand the cellular composition of the blood. Blood consists of fluid called plasma and formed elements. The formed elements include erythrocytes, leukocytes, and platelets. This figure includes neutrophils, eosinophils, basophils, monocytes, macrophage, dendritic cell, lymphoid stem cell. Neutrophils, which are highly phagocytic and motile, are active in the initial stages of an infection. Eosinophils are somewhat phagocytic and also have the ability to leave the blood. The major function is to kill certain parasites. Basophils release substances such as histamine that are important in inflammation and allergic responses. Monocytes are not actively phagocytic until they leave circulating blood, enter body tissue, and mature into macrophages. Dendritic cells are especially abundant in the epidermis of skin, mucous membranes, the thymus, and lymph nodes. Dendritic cells destroy microbes by phagocytosis and initiate adaptive immune responses. Lymphocytes includes natural killer cells, T cells, and B cells, destroy target cells by cytolysis and apoptosis. Phagocytosis is a process in which certain cells in the body devour and destroy microorganisms. How does phagocytosis occur? For the convenience of study, we will divide phagocytosis into four main phases: chemotaxis, adherence, ingestion, and digestion. Chemotaxis is the chemical attraction of phagocytes to microorganisms. Adherence is attachment of phagocytes plasma membrane to the surface of microorganisms or other foreign material. Following adherence, ingestion occurs. The plasma membrane of the phagocyte extends projections called pseudopods. Once the microorganism is surrounded, the pseudopods meet and fuse, surrounding the microorganism with a sac called phagosome, or phagocytic vesicle. Next, the phagosome pinches off from the plasma membrane and enters the cytoplasm, where it contacts lysosomes that contain digestive enzymes and bactericidal substances. The contents of the phagolysosome brought in by ingestion are digested in the phagolysosome after enzymes have digested the contents of the phagolysosome brought into the cell by ingestion. The phagolysosome contains indigestible materials and is called a residual body. The residual body then moves toward the cell boundary and discharges its waste outside the cell. Although some pathogens can cause damage on the surface of tissues, most must penetrate tissues to cause disease. Here we will consider several factors that contribute to the ability of bacteria to invade a host. Some bacteria make glycocalyx material that forms capsules around their cell walls. This property increases the virulence of pathogenic species. The capsule resists the host's defenses by impairing phagocytosis, such as Streptococcus pneumonia. The cell walls of certain bacteria contain chemical substances that contribute to virulence. For example, Streptococcus faegens produces a heat-resistant and acid-resistant protein called M protein. Microbiologists think that virulence of some bacteria is aided by the production of extracellular enzymes and related substances. The fibrin clot may protect the bacterium from phagocytosis and isolate it from other defenses of the host. Coagulases are produced by some members of the genus Staphylococcus. Once inside the host cell, certain bacteria such as Shingilla species and Listeria species can actually use actin to propel themselves through the host cell cytoplasm and from one host cell to another. Still, microbes have the ability to survive inside phagocytes. Shingilla and Rixta species have the ability to escape from a phagosome before it fuses with a lysosome. HIV can prevent the fusion of a phagosome with a lysosome and the proper acidification of digestive enzymes. Damage to the body's tissues trigger a local defensive response called inflammation, another component of the second line of defense. Inflammation has the following functions: to destroy the injurious agent, if possible, and to remove its byproducts from the body. If destruction is not possible, to limit the effects on the body by confining or walling off the injurious agent and its byproducts, and to repair or replace tissue damaged by the injurious agent or its byproducts. During the early stages of inflammation, microbial structures such as flagellin, LPS, and bacterial DNA stimulates the toll-like receptors of macrophages to produce cytokines. The liver synthesizes a group of proteins called acute phase protein. Kidneys are another group of substances that cause vasodilation and increased permeability of blood vessels. Kidneys are presented in blood plasma, and once activated, they play a role in chemotaxis by attracting phagocytic granulocytes, chiefly neutrophils, to the injured areas. Various components of the complement system produced by the liver stimulate the release of histamine, attract phagocytes, and promote phagocytosis. Active fixed macrophages also secrete cytokines, which bring about vasodilation and increased permeability. Vasodilation and the increased permeability of the blood vessels also help deliver clotting elements of blood into the injured area. We will divide the process of inflammation into 3 stages: vasodilation and increased permeability of blood vessels, phagocyte migration and phagocytosis, and tissue repair. Immediately following tissue damage, blood vessels dilate, increase in diameter in the area of damage, and their permeability increases. Dilation of blood vessels, called vasodilation, is responsible for the redness, edema, and heat associated with inflammation. Generally within an hour after the process of inflammation is initiated, phagocytes appear on the scene. As the flow of blood gradually decreases, phagocytes begin to stick to the inner surface of the endothelium of blood vessels. This sticking process in response to local cytokines is called margination. Then the collected phagocytes begin to squeeze between the endothelial cells of the blood vessel to reach the damaged area. The phagocytes then begin to destroy invading microorganisms by phagocytosis. Certain chemicals attract neutrophils to the site of injury, chemotaxis. After granulocytes and macrophages engulf large numbers of microorganisms and damage tissue, they themselves eventually die. As a result, pus form, and its formation usually continues until the infection subsides. As effective as phagocytosis is in contributing to innate resistance, there are times when the mechanism becomes less functional in response to certain conditions. For example, with age, there is a progressive decline in the efficiency of phagocytosis. Recipients of heart or kidney transplant have impaired innate defenses as a result of receiving drugs that prevent the rejection of the transplant, and so on. The final stage of inflammation is tissue repair, the process by which tissue replaces dead or damaged cells. This is the process of inflammation. A. Normal blood flow in the tissues as injury occurs. B. Substances released cause dilation of small blood vessels and increase blood flow in the intermediate area. Phagocytes attach to the endothelial cells and then squeeze between the cells into surrounding tissue. D. The attraction of phagocytes causes them to move to the site of damage and inflammation. Collections of dead phagocytes and tissue debris make up the pus often found at the site of an active inflammatory response. While inflammation is a local response of the body to injury, there are also systemic or overall responses. One of the most important is fever. An abnormally high body temperature, a third component of the second line of defenses. The brain's hypothalamus is sometimes called the body's thermostat, and it is normally set at 37 degrees. When phagocytes ingest gram-negative bacteria, the LPS of the cell wall are released. LPS causes the phagocyte to release the cytokines interleukin-1, IL-1, along with TNF-alpha. IL-1 is an endogenous protein. These cytokines cause the hypothalamus to release prostaglandins that reset the hypothalamic thermostat at a higher temperature, thereby causing fever. The body will continue to maintain the higher temperature until the cytokines are eliminated. The thermostat is then reset to 37 degrees. As the infection subsides, heat-losing mechanisms such as vasodilation and sweating go into operation. This phase of the fever, called the crisis, indicates that body temperature is falling. Up to a certain point, fever is considered a defense against the disease. A moderate fever is good for the body. It can inhibit pathogen growth and increase cellular metabolism. IL-1 helps set up the production of T cells. High body temperature intensifies the effect of antiviral interferons and increases production of transferrins that decreases the iron available to microbes. Also, because the high temperature speeds up the body's reactions, it may help body tissue repair themselves more quickly. However, high fever also had a bad effect on the body. When the temperature is higher than 40.7 degrees, it may be dangerous, which can cause tachycardia, acidosis, dehydration. As a rule, death results if body temperature rises above 44 to 46 degrees. Thank you for your attention.